Uh, First lecture was on polynucleotides. Now I come here today with the second one, where we will go into more of details of deoxyribonucleic acid. Now deoxyribonucleic acid, it is a double helical structure. We call it as a double helical structure. What does that mean? When two polynucleotides come to lie side by side with the bases, with the nitrogenous bases of both the strands projecting inwards, that means in the same facing each other, the bases, they face each other of the two strands, and they get bonded to form they get bonded with the help of hydrogen bonds, thereby remain attached together. That makes it double stranded, and both the two strands, then they twist, giving a in the form of a helix. So we call it as helical structure. Like you can see here, it can be seen here that the two polynucleotides, the both of these two, which appears like a ribbon, which appears like ribbons, these are two strands with with bases of both the strands projecting inwards. They're projecting inwards. This is A, G, then G, then A. Okay, the basis of this first strand I'm talking about, A, then again furthermore here A, C, A, then furthermore G, T, C. So these are the bases of one strand. The other strand also accordingly, it lies parallel to it. Parallel to it, it lies on its side with the bases projecting towards the base of the first strand. They come quite close to each other in such a manner that they form hydrogen bonds. This is facilitated due to the molecular structure. They get bonded to each other, thereby both the two strands remain together, attached together. At the same time, what happens? They will turn in the form of an helix. So we call it as double helical structure of DNA. For the first time, it was proposed by James Watson and Francis Crick in 1953. Now, another scientist, Chargaff, put forward a rule, put forward a sort of rule, a hypothesis telling that how the formation of helix is possible, how the formation of this two-stranded structure is possible. What enables this formation? His rule states that always pairing occurs between adenine and thymine, guanine and cytosine. As you can make out here, adenine and guanine both are purines. Thymine and cytosine both are pyrimidines. So pairing always occurs between a purine and a pyrimidine. At the same time, those are also specific. Adenine will pair with thymine, guanine will pair with cytosine. This stands true for all living organisms. So Chargaff stated this fact. And secondly, he stated that always there is double bond between adenine and thymine, double hydrogen bond, and triple hydrogen bonds between guanine and cytosine. Third point, which Chargaff stated, that the ratio of the total quantity or amount of adenine and guanine added together to thymine and cytosine added together is always equal. That is, which he gave as the unit value. So these were the points stated by Chargaff. So here we have a picture which is depicting all of these facts. Adenine pairs with thymine, okay, or thymine pairs with adenine. Both ways it is the same thing with double hydrogen bonds. Guanine cytosine, okay, cytosine guanine or guanine cytosine with triple bonds. They are bonded. So it gives us the double helical structure. At the same time also, if you observe it carefully, both the two strands, you can see here, okay, the position of sugar, it is opposite to each other. Okay, so if one of the strand is marked as 3' to 5', the other strand will be 5' to 3'. That means they are parallel They lie parallel to each other but in an anti-parallel manner. The position of the two polynucleotides is always anti-parallel to each other. Then the salient features of DNA: two polynucleotide strands joined by hydrogen bonds. I have already mentioned about this point. Complementary base pairing. Complementary base pairing means that adenine pairs with thymine and guanine pairs with cytosine. So this is known as complementary base pairing rule. So here, this is the complementary base pairing rule: TAAT, GC, here also GC, CG. So this is the complementary base pairing rule, and they are held together by hydrogen bonds. Then they are always coiled in a right-handed manner. The two strands are anti-parallel. Okay, the two strands are always, so here you can see they are coiled with an imaginary axis. If you place here, they are coiled in a right-handed manner. This gives us the helical structure. So sugar phosphate backbone, okay, they are lying outside. Here also it's outside, and with the bases projecting inwards. The nitrogenous bases, as pointed out here, adenine, thymine and guanine, cytosine. They will be held together by hydrogen bonds. Then the polarities of the two strands, as already mentioned in the previous slide, 5 to 3 and 3 to 5. Two base pairs. Now this is one important point here. The distance between any two base pairs, like say the distance between this first base pair and the second base pair, we call these as base pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9 base pairs is making up this small portion. So distance between any two base pairs is 0.34 nanometers. Okay, it's 0.34 nanometers. And one complete turn means from here when it starts and it reaches the same position. Okay, that one complete turn is made up of 10 base pairs. It is made up of 10 base pairs. So we can estimate the distance covered in one turn. Okay, so if the distance between two base pairs is 0.34, so it's quite obvious 3.4 nanometer is the distance of one turn. So after every 3.4 nanometer, there will be one turn or one complete coiling of the strand, DNA strand. So here we can see the distances they are shown here: 0.34 nanometer between two base pairs, and one complete turn is 3.4 nanometers. Also, the distance between two strands here, it's 2 nanometers. The polarities, as you can see here, okay, the polarities as you can see here, this is 5 to 3, and this is 3 to 5. So they have to be in opposite directions, they move. So it is furthermore repeated. Okay, 3 prime. Why this end is known as 3 prime? Because it has the third position of carbon free, and this side the fifth end, that is with the OH groups, they are free. So here this is 3 and this is 5 end. So if one of the strand is here 3 to 5, the other strand has to be 5 to 3, with each nucleotide carrying one base. If this side a thymine, then the other side definitely a nucleotide carrying adenine has to be approached or has to come near thymine. Only then the bonding will take place that will enable the formation of the double-stranded structure. Such formations occur during replication process of DNA, which we will learn in the coming lectures. Then again, structure of DNA, the same things are repeated so that the picture remains in your mind. So here adenine thymine. So if one is moving in one direction, the other moves in the opposite direction. But both of them have to lie parallel. But what do we use the word? We use the word anti-parallel. Now next we will come to packaging of DNA. Now it is said, okay, it is said that the mammalian DNA itself is 2.2 meters long, which is enclosed within the nucleus of a cell. The cell itself is so very minute, and inside the cell, the nucleus, the DNA is enclosed. Now, how is that possible? That is possible because the packaging mechanism of DNA is quite unique. So here first we will learn about the prokaryotic DNA packaging. Now prokaryotic DNA packaging, okay, what happens? Prokaryotes, they do not have a nuclear membrane. In them, they have a single chromosome which simply lies within the cell. It simply lies within the cell. So here what happens? The DNA, its loops are formed. Okay, they get folded upon each other. Okay, they get folded in this manner. They get folded in this manner with the presence of some positively charged proteins in between. DNA is negatively charged. Why? Because it has carboxyl group at the fifth position, phosphoric acid. It makes it negative in charge. When with the presence of positively charged histones, what happens? The negatively charged DNA gets coiled or gets wrapped around the positively charged proteins, as you can see here. Okay, these are the positively charged proteins around which the DNA gets coiled. Due to opposite forces, opposite charges, what happens? They can remain together. So consequently, what happens? The structure gets compressed. The whole DNA structure gets compressed. Furthermore, they form loops, as you can see here. Okay, this is the first structure. Then furthermore, what will happen? They will form loops. Okay, these are the loops, and that will again further coil. They will uh, be folded. Okay, they will form a loop structure, and this is the loop structure. So here when we see the chromosome, we will get this appearance. Further, if we try to peep into it, what is present there in one loop is this. So from here when we try to see, then we will get to identify that it has a positively charged protein around which the negatively charged DNA is coiled. So in prokaryotes, this structure is also known as nucleoid. Okay, it is to be because they do not have a nucleus, proper nucleus, so we prefer to call it as a nucleoid. So negative DNA coiled with the positive proteins in loops. Then in case of eukaryotes. Now in case of eukaryotes, what happens? The eukaryotes, we have a proper chromosome. So this is the proper chromosome which we have. This is the chromosome. So the chromosome is made up of DNA. When we try to see what is present here, then it is seen that they're very highly coiled. Very, they're coiled. Then the supercoils. These are the supercoils. When we try to open up the coils, further we open up the coils. So you can make out here, when we open up the coils, we can find some clusters here. These are the clusters. Further, when we open up the clusters, it is seen that they are made up of some ball-like structures. These are the ball or bead-like structures. When we further try to open up the bead-like structures, we get the DNA. So what does it mean? Now I'll go the reverse direction. DNA gets enclosed or gets coiled around some positively charged histones. Positively charged histones. Why are they positively charged? Because they have abundance of amino acids, lysines and arginines, okay, which are positively charged amino acids. So the proteins that are made with abundance of such proteins, consequently what happens? The protein itself is positively charged. It turns out to be positively charged. So those are known as histones. Around these histones, the DNA gets coiled. It gets coiled, it forms a structure which we call as nucleosome. These nucleosomes, further, okay, what will happen? Further, they will undergo coiling. Further, they will undergo coiling. Okay, more and more coiling, and finally, it gets so very compressed, we get the structure of chromosomes. So in eukaryotes, let us see. Okay, that was the figure which I showed. Now let us see what happens. It is also known as beads on string. The structure is also called as bead on string. Histones, as already mentioned, positive basic proteins due to abundance of amino acids, arginine and lysine. Now, arginine and lysines, due to their abundance, some polypeptides are formed, that is, proteins are formed. Those proteins are positive charged, called as histones. The histones, now again, 8 histones, they come together which we call as octamers. This octamer of histone referred to as nucleosome. So the octamers, they can be, or the histones can be classified as H2A, H2B, H3 and H4. These four types, they exist in pairs. Each of them exists in pairs that counts to total number 8, making an octamer. In addition, they have one more, that is H1. But H1 does not form a part of the octamer. It lies outside to the octamer. So we keep it separate. Then there are it has been seen that around each nucleosome, 200 base pairs coil around one nucleosome. 200 base pairs. Okay, so total number of base pairs, we can even count them, like say distance between two base pairs is 0.34 nanometer. So 0.34 nanometer into 200, we can take the value, the length of that much length of DNA will coil around one nucleosome. One nucleosome made up of these four types in pairs of histones. Repeating units of nucleosomes, they form the chromatin. So here we have the picture now. This is the cell. Okay, this is the cell. This is the nucleus. Nucleus has the chromosomes. So chromosomes, this is when we open it up, we get the nucleosomes, and finally we get the DNA. Now here, this one nucleosome, okay, here it's being shown. So you can see here there are 8, 1, 2, 3, 4, then below the 4. Okay, so these 8 histones, H2A, 2B, 3 and 4, they make up one nucleosome around which the DNA, this blue is the DNA, it gets coiled. Okay, it gets coiled. So you can see here 2.5 coils. Also it is referred to as 2.5 coils. Okay, it gets coiled. On the side is present the H1 histone. Okay, to prevent it from slipping down. Further, the same thing here, it has been enlarged. So the green things, these are the histones, 8, 1, 2, 3, 4 and below 4. 8 histones around which this red one is the red, the thread-like structure is the DNA that is shown here. It coils and on the side is present H1. So such structures, numerous, all these black dots that you can see, they are the nucleosomes. These nucleosomes will again aggregate. 10, usually 10, they will again come together. That 10 will further coil. Again it will coil. 10, 10, 10, they will aggregate. So that further supercoiling and coiling will result into a structure which will be very, very compressed. Then there are, besides this histone, some non-histone chromosomal proteins are also, besides the histones that are present inside the nucleus, helps in aggregation of the DNA. There are some non-histone chromosomal proteins also. Non-histone, okay, they are not the same composition as the histones. They are known as non-histone chromosomal proteins, NHC. So here you can see they are the nucleosomes, okay, which are formed of histones here with the DNA coiled around them. Then the second nucleosome, the third nucleosome. Then in between these green dots that you see, green ball-like structures that you see, these are the non-histone chromosome proteins, which helps in additional attachment of one nucleosome with the second, second with the third, and third with the fourth, so that they can further and further they get compressed. Furthermore, the entire chromosome, okay, the chromosome, if we stain the chromosome for identification, as you are familiar, for identification of any cell or any cell component, what do? We use stains. So when we stain to study chromosomes, it has been observed that a particular portion of or some regions of the chromosome, they stain very light, whereas some they stain very dark. The ones which are lightly stained are called as euchromatin, and the ones which are darkly stained are referred to as heterochromatin. The euchromatin, further, when it was seen, like why does it stain light? It was observed that they are little slightly loosely packed. The packaging system is loose. Packaging means this how the nucleosomes are attached. Okay, so it's slightly loose, whereas the heterochromatin, they are more dense, they are more tightly packed. That is why we see them as dark. That portion appears dark. Furthermore, it was also seen that the portion which is stains light and is loosely packed, they are the active portion of the DNA, whereas the ones which are tightly packed are inactive portion of the DNA. They do not participate in formation of any proteins. They do not participate to function as a gene, whereas the euchromatin portion, which is the active portion loosely packed, they are found to be involved in expression of characters in the individuals. So chromosomes, the chromosome itself, we can categorize it as euchromatin and heterochromatin. And please take note of this point that we are talking of the same chromosome. Single chromosome, a portion may stain light, a portion may stain dark. The lighter portion is more active, the darker portion is inactive. So this is about the packaging of DNA. The next lecture series, we will further move into the more deeper concepts.